Before talking about Prolyl hydroxylases, also known as PhDs, I would like to give a brief introduction about hypoxia. So, as probably some of you already know, hypoxia is the condition in which cells experience a lack of oxygen. And when that happens, an hypoxia response is triggered so that cells can try to survive to such a condition. And fundamental for such a response are the HIF factors that are heterodimers. So there are two subunits, a subunit alpha and a subunit beta. And under hypoxic conditions, HIF dimerizes, and the dimer itself binds to the DNA to start the transcriptions of factors that are fundamental for the cell survival. There are 3 isoforms of the subunit alpha, whereas there is just 1 isoform of the subunit beta. The main difference in between the 2 subunits is that the subunit alpha have cod and nod domain, also known as oxygen degradation domains. And the reason why I'm mentioning these domains is that they are important for what comes next. So the HIF factors are constitutively expressed in cells, which means they're expressed also when they're not needed. Therefore, they need to be degraded. And this is when PhDs come into account. So PhDs enzymes are responsible of the hydroxylation of the HIF alpha factors on proline residues that are located in nod and cod domain. Once the hydroxylation occurs, the hydroxylated HIF alpha can recreate a complex that's called PBHL, and this is going to start the proteasomal degradation of the HIF alpha factor. So the PhD enzymes are fundamental for the downregulation of this factor. The PhD enzymes are part of the 2G oxygenases family. These enzymes are found in many different pathways like antibiotics biosynthesis, DNA repair, and oxygen sensing, which is what we are talking about today. In humans, there are 3 isoforms, PhD2, 1, and 3. They all are oxygen-censored, 2G and oxygen-dependent, and iron-dependent. The oxygenase domain is fairly conserved amongst these enzymes. What changes is that they are differently expressed in different tissues. And today I'm going to be talking mainly about PhD2, which is it's considered the most important human oxygen sensor. So the answer I'm looking for is, what is the mechanism of the hydroxylation reaction that occurs in between PhD2 and HIF alpha? And I'm trying to answer this question using time-resolved crystallography that basically allows us to take snapshots of this reaction. And I'm also trying to explore the role of different residues both in the active site and in the near proximity. And this hopefully will also help characterizing oxygen channels in the protein. So there is a commonly accepted mechanism which suggests that the enzyme in the resting state has an iron to the active site that's coordinated to a molecule of water and 2G, and the proline of the substrate is exposed to the active site. When there's oxygen in cells, what happens is that the 2G gets decarboxylated to form succinate, and the proline residue gets hydroxylated, and in the proposed mechanism, what's suggested is that ferrite complex forms. So how do we study this mechanism? What do we need to do in order to characterize this reaction? So the first step is to obtain a crystal structure of the starting complex of the enzyme in the resting state. So what I needed to do was to find out some anaerobic conditions to crystallize such a complex. The next step is to prove that this complex, these crystals can be exposed to oxygen, and hopefully prove that these crystals are also catalytically active. so that then we can harvest crystals at different time points to hopefully see different steps of this reaction. So after a lot of effort, we obtained the first anaerobic crystals of what I thought would have been PhD2 in complex with substrate 2Ag and iron. Funnily enough, the first structure we obtained was actually, I read the surprise, I'm sorry. So the first structure we obtained was an empty structure. By empty, I mean there was no iron in the active site, nor 2G. In pink here, I'm representing the amino acids that are meant to coordinate the iron, and this is a piece of the substrate. So the code domain of one of the Hif-alpha factor, and this is the proline that's meant to react. So a bit of more optimization of this gave us the structure of the first resting state complex. So this time you can see 2G is binding the iron in a bidentate manner. There's a molecule of water coordinated there, and the proline of the substrate is still not hydroxylated, which proves that our conditions were actually anaerobic. So at this stage, well, what's interesting is that the resting state seems to be in accordance to what's the proposed mechanism. So the next step was to prove that these crystals could be exposed to oxygen without them getting dissolved, and try and see if these crystals were actually active. So holding our breath, we exposed these crystals to oxygen, hoping not to see them melting away. And they were actually surprisingly robust. So we managed to harvest some of them at different time points, so with different oxygen exposure time. And we observed some hydroxylation in the active site, so there was actually turnover. So here you can see the 2G is being turned over to succinate, and the residue, the proline residue, is being hydroxylated. Interestingly, we can see two conformations of this residue, and we can also see an extra density on the iron that we are not able to characterize at this stage of the research. Although this seems to be promising for future studies. Longer time exposure let us observe how the proline arrives at the final conformation, where it's fully hydroxylated, and iron and succinate are leaving the active site unexpectedly, whereas the product stays coordinated to the protein, although it's worth mentioning that probably this is a crystallographic artifact. So one of the main problems with these experiments is that they've all been conducted in cryo conditions, so we have a temperature bias in there. Also, crystals were harvested manually, which means it took us minutes before we could harvest them. So the reaction was already started, so this way we sort of missed part of the reaction. So we started to think about conducting time-resolved experiments using XCell. So XCell stands for X-ray free electron lasers, and there are synchrotrons, actually there are 5 synchrotrons around the world that allow users to use XCell beamlines. And the main characteristic of these beamlines is that you can expose crystals to high intensity beam in the X-ray regime, and this can be done using pulses duration. So basically, the crystals can be exposed very fast to high intensity beam. So this allows us to monitor reactions that occur really fast. Also, we can use, the experiments are conducted at room temperature, which removes the temperature bias that we might have with cryogenic conditions. Due to the high intensity of the beam, we cannot work on single crystals, but we need microcrystals because the crystals are destroyed in the moment they are hit by the beam. So the first step was to optimize microcrystals. Here's just a picture to show what's the difference in dimensions. And the first experiment we conducted was a Prolyl cell, where we just sent the first microcrystal slurry obtained to check whether or not this crystal is destructive. In fact, one of the main problems with small dimensions crystals is that reduced dimensions might also mean way reduced resolution, which is a big problem when we're studying mechanism. So the experiment we did at Prolyl cell is a fixed target experiment where we just exposed our microcrystal slurry to the beam using a chip. So basically, the microcrystal slurry is deposited in between two polyimide films, and everything was at room temperature, but also everything was aerobic, which means all we could get was a product structure, if anything. And we were quite lucky. We obtained a room temperature structure of the product in context with our enzyme. What this experiment told us was, first of all, we could actually use those microcrystals for more complete experiments, like oxygen exposure experiments. And also, the RMSD of this structure when compared with the prior structure was quite low, which means there are possibly no big conformational changes when we are at different temperatures. So the prior structure might not have any temperature bias. So the next experiment we thought to conduct was LCLS, which is part of the SLAP facility. So at LCLS, it is possible to collect data in anoxic conditions, and it's also possible to expose the crystals to oxygen in a controlled manner. The way the system works is, there should be a video coming up soon. So a microcrystal slurry is deposited on a tape, and this tape moves in a chamber that can be either anoxic or at a specific concentration of oxygen. Every droplet contains a certain amount of crystals, and it's taped by the beam. And by the end of the experiment, all the data obtained are merged to solve a crystal structure, hopefully. The only experiment we managed to do when I went a few months ago was an anoxic experiment. So, and what we obtained was a crystal structure, again, of the resting state of the enzyme, which seems to be really similar to what I was saying with cryogenic conditions. So, just a brief summary of what's been obtained so far. We obtained very reproducible anaerobic crystallization conditions, and we were able to characterize the starting point of the reaction and the end point of the reaction. We also managed to optimize microcrystals conditions for Excel studies, and we removed the temperature bias out of our data. What we are aiming to do next is to expose our crystals to oxygen at different time points. Although there are many challenges when it comes to this experiment. First of all, our enzyme has high oxygen KMs, since they are oxygen sensors. This means that oxygen exposure might be a failure, because the oxygen might diffuse too slowly through our drops. On the other hand, if the oxygen diffusion is not a problem, the other challenge would be to isolate the intermediates, because they might either form or decompose too fast. Although there is a small solution to this last challenge, which would be co-crystallize protein with different substrate and cofactors to hopefully slow down the reaction, and this is something we are currently doing. And this brings me to the acknowledgements. So I would like to thank my supervisor, Chris Cocker, for having me on working on this project, all the people involved, all the synchronomes that gave me being time, and the medical sciences for funding my studies, and Instruct-ERIC for having me here today. Thank you.